So we're going to move on to our next speaker, who is Nicolas Pedroncelli. And so Nicolas graduated from UCLA with a bachelor's degree in bioengineering in 2022. And since graduation, they've been a research associate at A2 Biotherapeutics, advancing the next generation of CAR T therapies for solid tumors. And their research focuses on synthetic biology and tissue engineering. So I'll let you go ahead and share your slides and take it away.
Awesome. Thanks for having me. While I'm pulling up my screen, just want to say thank you for this opportunity. As I said, I'm no longer an undergraduate. I've been working in research for about a year now during my gap year, but I am applying to biomedical engineering graduate programs right now. And I came across Paper Trails on Twitter and replied in the thread asking, I'm no longer an undergrad. Am I still able to apply? And I was met with open arms and so applied, and I'm here now. And so again, really grateful for this opportunity to still share my research and get some practice in science communication before actually going to grad school. So let me pull up my screen real quick. I'm having a little bit of difficulty with PowerPoint, but give me 1 second. Okay. Is this showing the full screen or presenter view?
The full screen, you're good.
Okay, cool. Alrighty. Yeah, so I graduated. My name's Nico. Graduated from UCLA last year, worked in the Song Lee lab. And so today I'm going to talk a little bit about my research that incorporated synthetic notch signaling within 3D microtissues to try and orthogonally control cell differentiation and better form tissues in vitro. So to start things off, it's important to kind of set the stage that tissue function and their complex architecture are inherently very intertwined. So to give a couple different examples of this, if you look at the kidney, multiple different regions and these complex tubule structures allow it to form these gradients that are necessary for its filtration mechanisms. If you look at skeletal muscle tissues, the innervation of neurons and the precise alignment of these myofibril structures allow for it to have the coordinated contraction, and that results in our movement of all the different muscles. And then if you look at across all our different organs and tissues, there's this very complex, pervasive vasculature that is intertwined all throughout, and that, as we know, allows for the delivery of waste and oxygen. So going back to what Dr. Smith had posed earlier, there's this organ shortage crisis, we want to be able to create these organs either for regenerative purposes or for replacement purposes. This complex architecture kind of poses itself with how would you even go about engineering these complex tissues? They're so complex, where would you even start? So that brings us to the strategy of tissue engineering, which Dr. Smith also alluded to or showed on one of his slides, but we have this tissue engineering strategy, which is based on this fundamental idea of combining the components of tissues, mainly different cells, signaling molecules, onto some sort of biomaterial scaffold, and kind of hoping that biology takes over and you can create these tissues of all these different components from a bottom-up approach. So one example, when you're trying to, let's say, create an artificial skeletal muscle in vitro, you could take some pluripotent stem cells, you can differentiate them into all the different cell types with all involved in skeletal muscles. You can then further mature them and then combine them, all these different components, onto biomaterial such as fibrin. And again, kind of cross your fingers, hope that everything kind of organizes on its own together, and hope that this all results in this in vitro skeletal muscle for disease modeling or tissue replacement strategies. But as you can see here, this multicellularity, there's a lot of different components. It's very challenging and tedious to engineer, especially the individual components that they're all spatially coordinated. So my battery's running low. Let me plug in my laptop real quick. So that brings us to the point of how can we better achieve this process? So my project, it's specifically focusing on incorporating synthetic cues to try to jump this middle pathway and go directly from a more naive cell state and jumping this developmental biology platform to try to synthetically or incorporate synthetic biology to more efficiently fabricate these complex tissues. to again try to get back at that better creating tissues for organ replacement or for regeneration. So the synthetic biology tool that I use in my project is called synthetic notch or synotch signaling. And this is based on the native cell communication mechanism called notch signaling. Excuse me. And how that works is you figure here on the left, when a sender cell that presents this delta ligand binds to the notch ligand of a receiver cell, this results in the exposure of a cleavage site. But then a protease or an enzyme can cleave this intracellular domain, which results in the trafficking of a transcription factor, for example, and then the specific expression of notch-related genes from the binding of these two cells. So the synotch mechanism was developed a little under a decade ago, and how it works is the researchers, you could just replace the intracellular and extracellular domain to use the same force-induced cleavage mechanism to result in a novel transcriptional output or customizable gene expression from any novel environmental input. So again, replacing this notch and delta combination with any recognition and effector domains. And synotch has been used for a wide variety of applications. But what my project specifically is focusing on is trying to use synotch to induce differentiation and thus create more complex tissues in vitro. So the cell line or the synotch pathway we used was recently validated in a preprint where they showed that from a GFP synotch activator, you can actually transdifferentiate fibroblasts into muscle cells by GFP activated MyoD expression. So how this works is over here in the top left. Here's a schematic of the cell line. So again, when GFP binds to this synotch cell, that results in this cleavage of the synthetic transcription factor, which is when trafficked to the nucleus to this binding site, results in the precise expression of MyoD, which is a muscle lineage transcription factor to drive the expression of muscle-related genes and try to transdifferentiate these cells, and also M-cherry, which is a red fluorescent marker. So just to go over a little bit of the data real quickly from the paper, they show that this cell line was able to differentiate into muscle cells from GFP, specifically looking at this positive alpha actinin, which is a marker for muscle cells. And you can see that there's positive staining in the presence of GFP, but also we see the presence of multinucleated cells, which is indicating the fusion of these cells into myotubes or muscle-like fibers. We also see there's a very highly aligned alpha actinin pattern. So overall, this cell line was validated to be very GFP activating specific to differentiate into muscle cells. And so I worked on this a little bit as an undergraduate researcher in the Lee Lab and was included as a co-author on this project. But I also had another project in the lab where we were asking the question, how can the cell line be expanded into other tissue engineering systems? So that brings us to a 3D culture device that I helped develop within the Lee Lab. This was initially used, initially a capstone team project we had my senior year to use as a platform to assess microneedle delivery that were being developed in the Lee Lab. So how these work, here's a little video from literature from a seminal paper a little over a decade ago that shows in these PDMS, so this rubber-like material, if you create these devices with these pillar-like objects, pillar-like structures, and you seed a collagen or seed a hydrogel mixed with different cell types, over about a 24-hour period, you see this remarkable self-assembly of the structure around these pillars, and you can see it's forms into like a tissue-like structure that has relevant mechanical forces, and you can actually measure the maturity or the contractile properties of this tissue that's in suspension just based on the deflection of these pillars. So in the Lee Lab for my capstone project, we developed MilliMuscle, which is a PDMS device and a myoblast collagen hydrogel that was a little larger than previous microtissue devices, and that's to, again, have it on the scale for the lab to assess their microneedle delivery strategies that they've been developing. So develop this PDMS device. I could talk a little bit more about it later if anyone's interested, but essentially what we did in this project, we created this device and showed that we were able to create these mini muscle microtissues in a pretty reproducible fashion. So that kind of brings us back to synotch. And where my project came in is how can I kind of combine this capstone device with my undergraduate research project using Synach. So what we did is we combined these Synach senders and receiver cells. So these senders being GFP presenting, that's that active Synach activating ligand, which with the Synach receivers. So the Synach receivers are the cell line that expresses MyoD and M-cherry in the presence of GFP. So we combine that with a type 1 collagen-based hydrogel. We seeded that into the Millie muscle culture platform. And what we were really asking is, can these fibroblast Synach cell lines, in the first place, can they even self-assemble? Can we even see robust tissue formation out of them without muscle tissues or without muscle cells? Do we see Synach activation? Do we see any M-cherry signal within these 3D microtissues? And how might this MyoD expression, how does this induced Synach activation affect cell differentiation or affect the tissue phenotype or maturity? So this is hasn't really been explored yet in literature. There's been a lot of Synach applications in material activation or 2D substrates, but there hasn't been much specifically in the microtissue space. That's why we asked the question: is this even possible? So it's very exploratory. Didn't really know what we were looking at or looking for. So what we found is after 24 hours, the Synach cells recapitulated this microtissue self-assembly. So you see here on the left, tissues made from both Synach receivers only. So that's the MyoD M-cherry line, as well as a co-culture of the receivers and senders. Both were able to reproducibly form tissue. So here's just a bright field representative images of this. And then we see in the bottom right, we actually see a slight uptick in M-cherry expression. So this would be that fluorescent reporter indicating Synach activation. And we only see that in the presence of this GFP input, since this is the co-culture with the sender cells. And then in the top right, we don't see any M-cherry signal. And that's because, again, this is Synach receivers only. There's been no GFP input here. So this is a good preliminary example that Synach might actually be activating in a GFP-dependent manner within this construct. Then after day 3, we see robust GFP-dependent Synach activation. Again, looking here in the bottom right, we see a really robust M-cherry expression throughout the tissue that definitely correlates and overlaps with the presence of GFP. And still on day 3, we see no GFP-independent activation of M-cherry. So from here, we confirm that Synach is able to activate within these tissues, which is a very exciting observation, again, because this is very exploratory and did not know how Synach would behave in a 3D tissue environment. But more exciting result we saw on day 10, we started to see the appearance of aligned Synach activated cells into what resembled these muscle-like fibers. So on day 10 of this tissue, so this is a tissue with both the GFP sender cells and the Synach cells. If we zoom in, by day 10, we actually start to see very aligned structures of this M-cherry signal. So these are the Synach activated cells. And this alignment actually resembles some established muscle microtissues seen here in the bottom right from literature. So this is a super promising result, and we were really excited by this, that indicated the potential Synach-induced phenotypic change from this MyoD expression. So, not only was Synach activating in these tissues, but the phenotypic change of these structures and the development of these muscle-like alignment was indicating that the MyoD expression was actually having an effect on the cell differentiation and then subsequent tissue maturation, which was super exciting to see. So just to kind of wrap things up, what this project was based on was overall trying to expand this tissue engineering toolkit with synthetic biology. So to kind of just recap what I had done in my research project, I incorporated an inducible Synach cell line into traditional PDMS-based microtissue systems, which still hasn't really been explored in literature. Also showed proof of concept for muscle tissue formation via this orthogonal Synach signaling from MyoD expression. After I graduated, there still need to be some experiments needed to validate and optimize this cell differentiation and really assess the phenotype and maturity of these tissues. So there's definitely more staining experiments that could have been done to get at what the actual phenotype of these cells were within these muscle tissues or within these microtissues. And since then, the graduate student I was working with has been adding other Synach cell lines into tissue engineering platforms, such as within endothelial chips to again, try to better guide tissue formation. which has been super exciting. But just for the sake of this project or sake of this presentation, I just focused on the data I had generated. But this work is being continued in the Lee Lab. So again, just to recap, the overall goal of this project would be to better spatially coordinate or generate complex tissue architectures in vitro. So the overall goal of this project would be creating a multi-lineage synapse cell line, which has been validated in that preprint that I discussed earlier. And if you bioprint different materials with these different synapse ligands, so GFP and RFP being that red and green, you can see that the GFP or the green regions would drive MyoD expression. So that would drive spatially coordinated muscle-like expression. Then the red regions would drive ETV2 expression, which is an endothelial transcription factor to try to create vasculature. So again, it'd be combining these multiple synapse components to try to create something that looks more like a tissue in vitro, and overcoming some of those developmental biology difficulties and using synthetic biology to try to jump that gap and expand the toolkit that we have. So just give a couple acknowledgements. I worked with a lot of different groups at UCLA. Really want to give a big shout out to Tyler Hoffman in the Lee Lab. He was my graduate student mentor that I worked with on all of these projects, and I still talk to him today, and he's been helping me a lot, both with this presentation and with grad school apps. And then everyone from the Morset lab who generated the synapse cell lines, my bioengineering capstone team, we worked on the device together, and then some funding I received for this project at UCLA. At that point, I'd like to open it up to any questions. Sorry, I think I went a little over on time.
Oh, no, that's fine. Thank you so much. That was such a great presentation. I love that thin notch system. So it's really cool to see someone using it. There's a question in the chat. So Chris, if
Yeah.
you want to ask the question yourself, you can go for it.
Hello.
Hey.
Fantastic talk. I joined partway through, so sorry if this was answered a bit earlier. But could you go back a few to where you're showing your red and green micro device?
Yeah.
Yes, right there. So my question is, could you chain multiple of these together to have a larger muscle system to be able to study multiple of these sections at once, to see if like longer sections with tension going straight across the pillars would improve cellular maturation?
Yeah, so that's definitely one of the ways the project was going. And you're looking at different dimensions of this PDMS pillar system, and that's kind of more on the capstone project. We made a couple different devices, and we're looking at how pillar dimensions and overall length and size of the tissue affects tissue maturation. Yeah, so I definitely think, if I actually go to this slide right here, the bottom right, this is a 2-pillar system, and that would drive kind of better alignment of these tissues. So I think overall, this would be a better design for this system. The 4-pillar system that we had used was, like I said, designed for the capstone project to look at microneedle drug delivery. So this was designed to better fit a microneedle size dimensions. But I totally agree that messing with the dimensions and kind of optimizing that would likely result in better muscle maturation.
Cool, thank you.
Yeah.
Great. And there's another question from the chat. I can just go ahead and ask it. So is there a need to optimize the pore size of the gel that you are including in your PDMS device? Would this optimization be specific to your application?
Yeah, that's a great question. So we weren't looking much at the physical structure of the gel. We were kind of using a pretty well optimized type 1 collagen hydrogel and simply mixing it with the cells and then seeding it into the tissue. We were looking at different effects such as like viscoelasticity and stiffness of different components on the synapse activation that I didn't really include in this presentation. But in terms of pore size, that's not something we ever looked into. Potentially, yeah, that could potentially help maybe with different nutrient diffusion, or maybe that would better promote myotube fusion and the fusion of cells into the myotube formation. So it's definitely something we could look at, but wasn't something we had ever really thought before. But I could definitely relay that message back to the lab. Thanks, John.
Great. Thank you so much. Thank you for sharing that work with us. And then I'll just ask you the final question of what kind of research are you interested in doing in the future? I know now you're doing some CAR T stuff. Is that something you want to continue as you apply to graduate schools, or is there another?
Yeah. So my undergrad was definitely focused more on tissue engineering with this little bit of synthetic biology spice I got from Synach, but working at the CAR T therapy company is a lot more on the synthetic biology realm. Like, how can we introduce different constructs to control cell signaling? So I've definitely garnered a better interest or a deeper interest or even skill set on that synthetic biology realm. So I'm applying to grad schools right now, primarily the bioengineering programs, looking primarily creating synthetic biology tools across a couple different disciplines. I'm still definitely interested in developmental biology. So a lot of my grad school apps are focused on creating synthetic biology tools to study things such as tissue patterning or morphogenesis. So that's kind of what I'm broadly interested in right now. I think I'll hopefully get into a school and find a lab that where I can pursue that. And so in terms of exact project, I'm not too sure. I just very fascinated by controlling cell behavior with different synthetic biology methods.
Great. Thank you so much.